Hi everyone, today I'd like to tell you about another coronavirus paper. This is an update to the previous two papers that we had discussed and following up on the story of recombination among coronaviruses and the potential role of recombination in the outbreak that we are currently suffering through. So this paper is brand new. It was posted to the preprint server bioRxiv just a couple of weeks ago. So it's brand new and has not yet been peer-reviewed or published. But I thought it'd be fun to tell you about it real briefly. So this is these authors analyzed a bunch of publicly available virus genomes. So there's no new experimental work and actually no new data. They just took data that's publicly available and analyzed it and came up with the results that are consistent with what we have discussed before with the two other papers, but it has a little bit more detail. So let's jump right to figure one. This is a figure that you will recognize from the two previous papers that we've discussed. From left to right on both of these graphs is the coronavirus genome from 0 to 30 kb. And these are two different visualizations of the different number of recombination events. So this is each recombination event plotted along the genome, and this is a sliding scale abundance graph. It's the same idea, and what both of them are showing is that in this gray region, which corresponds to the spike protein, so those are those little knobby things on the surface of the coronavirus that I'm sure you've seen in these images of the coronavirus that are being distributed. And then the greenish region within the gray region is a specific domain within the spike protein, which is known as the receptor binding domain. So this is the part of that spike protein that actually binds the receptor in the host cells. So a very important step of the infection. And you can see in both of these graphs that a lot of the recombination events that they detected comparing all of these genomes occur in this spike protein region, including the receptor binding domain, and this one in particular is showing that a lot of these breakpoints, and you can see it here, a lot of these breakpoints, so recombination literally involves breaking DNA molecules and then stitching them back together, so these breakpoints often occur just before this spike protein, indicating that the spike protein as a whole is being involved in these recombination events, which is what we had seen in the previous paper. And these authors see evidence for it as well. They also looked at recombination among MERS viruses. So MERS is a cousin of SARS, still within the beta coronaviruses, but causes a slightly different disease, and it shows up in a different place in the phylogenetic tree. And they don't have as much data from MERS as they have from SARS, but they do have several genomes from both humans and camels. The MERS outbreak actually came from camels, and they also see a lot of recombination events in the spike protein, and it's not quite as convincing as with the coronaviruses due to lack of data, but they do mention it here to say that SARS is not unique. Recombination also seems to be something that happens in MERS viruses, and perhaps especially in the receptor binding domain and the spike protein. All right, figure 3 is where it gets interesting. In this phylogenetic tree, I'll zoom in on, they are making a phylogenetic tree with the whole SARS coronavirus genome, and then they make a phylogenetic tree with only the receptor binding domain. So as we have seen before, when you make a phylogenetic tree with the whole genome, you get the new SARS-CoV-2 grouping separately from the first human SARS. So here's the first human SARS, here's the second human SARS that we are suffering through right now. They each have separate bat ancestors and different parts of the tree, right? And then if you make the tree with only the receptor binding domain, now the two human SARS viruses, which were previously separate, are now showing up in the same part of the tree. The first human SARS grouping more ancestrally than the most recent bat ancestor of the current human coronavirus, but separately from the other bat SARS-like viruses that were previously thought to be the ancestor of the human SARS virus. Not previously, but when you look at the whole genome, these bat SARS viruses are the ancestor of the human. When we only look at the receptor binding domain, the first human SARS looks more like the current human SARS, which again is what we saw in the previous paper that I discussed for you, which is that this receptor binding domain of the two human SARS viruses are quite similar, much more similar than you would expect from looking at the whole genome. So once again, consistent with what we've seen before. And the new thing that they have done here is then they plotted these recombination events and mutations along a timeline. So using the known dates of the first human SARS outbreak and the current human SARS outbreak, and some other molecular clock type of calibrations and extrapolations and interpolations, they estimate that the recombination events that led to the evolution of the current human coronavirus had to have occurred no later than 2009, which is when they predict the current human coronavirus diverged from its bat ancestor, and could have occurred as long ago as 1992, which is when they predict the first human coronavirus diverged from its bat ancestor. So these recombination events that we're talking about occurred sometime between 1992 and 2009, which is to say that the recombination events, at least those that are being focused on here with the spike protein, are not recent, and were not the recent events that triggered the current outbreak. These recombination events happened years ago and were already in some kind of coronavirus years before they caused the current outbreak. Now, we don't know if maybe it was already in humans and was just not causing an outbreak for some reason, or was in a different animal besides bats, but the recombination events are not that recent. They happened years ago. So that's kind of interesting. And then they go into a little bit more detail about how exactly they think this would have happened with this tree, which looks a lot like the previous tree, but now they're using this tree to formally propose what they think happened. So what they think happened is that there was a recombination event between 1992 and 2009 between an ancestor of the first human SARS virus and the ancestor of the current SARS virus, which almost certainly happened in bats. So there's a recombination event in bats during this time period between these two ancestors. And then subsequently, after the human lineage of the current coronavirus diverged from its most recent bat ancestor, there were several mutations in specific amino acid positions in the spike protein that occurred more recently. So it was actually these amino acid mutations, not recombination events, that may have been the straw that broke the camel's back, so to speak. They were the most recent things that happened before the outbreak. So that's interesting because a lot of people had been talking about a recombination event maybe being as the thing that triggered the outbreak, and there's actually no evidence for that. There's no evidence for a recent recombination event being the trigger. The more recent things that happened were these specific amino acid mutations, and at least one of those amino acid mutations is in a region that is involved in receptor binding domain, specifically the ACE2, which we had talked about before, and is the focus of a lot of treatment strategies being developed by pharmaceutical companies right now. So we still don't know exactly why this outbreak occurred. It clearly must have something to do with ACE2 binding, perhaps specifically these mutations that happened here. We don't know that. We don't know that for sure. There could be other mutations. There could even be another recombination event that people haven't really noticed yet, right? There's still a lot that's unknown, but at least right now the evidence is saying that recombinations happened long ago. Recombination was important for bringing the SARS-like receptor binding domain into this lineage, and then a bunch of more mutations had to occur before we got to the current outbreak. All right, that's it. Pretty brief paper, but pretty interesting, and it's a nice follow-up to what we had discussed before, so I thought you might find that interesting. Let me know what you think. See you later. Bye.